So what we're going to look at now is the structure of full protein molecules. So we know that protein molecules are polypeptides formed by lots and lots of amino acids all bonding together via amide linkages into a big long chain, a big long polymer. However, that big long polymer that we can call a protein has three different levels in its structure. The first level that we can look at of a protein molecule is its primary structure. So the primary structure of a protein molecule is simply the order of the amino acids that it consists of. So obviously there are lots and lots of different ways that there are about 20 amino acids that are very important in our bodies, and there are lots and lots of different ways that those and different orders that those amino acids can bond in a chain within our body. And so the primary structure of a given protein is the order that those amino acids bond in, and this is in fact dictated by our DNA. Our DNA dictates the order that the amino acids in our body bond in, so dictated by our DNA. Now, this primary structure, we have a very specific way of representing the primary structure. Each of the 20 amino acids that are present in our body, that are used for proteins in the human body, have evidently their own names. However, each of those names is shortened to a 3-character string. And so if we have multiple proteins, if we have multiple amino acids in a chain within a protein, then we can represent the primary structure by putting the 3-character strings of each of the amino acids in the order that those amino acids occur. So we'll go through that down here in our example in a bit, towards the end of this video. Now, the next part of the structure of a protein molecule is the secondary structure. Now, the secondary structure is formed by bonding or attraction between different parts of our chain. So often this will be due to hydrogen bonding between adjacent peptide or amide linkages. So that is also a very important thing. So what the secondary structure is kind of, maybe we'll have a string of proteins, but instead of having the string in a straight line, it will form maybe a coil or a spiral or some sort of helix shape. So if we have our protein string forming this kind of shape rather than a straight line, then we call that the secondary structure of the protein. So this is very often caused by hydrogen bonding between amide groups. Lastly, we have our tertiary structure. Now, the tertiary structure refers to the overall shape of the molecule. So although we may have our amino acid string curling like this, if we represent that coil as a straight line, then the tertiary structure shows how we may have it looping around on itself like that. So we may have a protein molecule that in fact has its string going in this shape. So that is the entire protein molecule, and the string curling and looping around itself seemingly quite randomly. And if we were to look closely at this string, it may in fact still be in a coil shape. So the tertiary structure is sort of the overall structure of the entire molecule and the way that it folds back on itself. And so this tertiary structure is caused by bonding or attraction between the Z group of the amino acids that form a protein. So we know that each amino acid has one carbon that has a carboxyl group coming off it and an amino group, and obviously the major group that dictates the behavior of the protein is the Z group, which also comes off that same carbon. So it's the bonding between the Z groups that dictates the tertiary structure of our protein molecule. Now, another very important thing to be aware of when it comes to proteins is that proteins can in fact act as markers for disease in the body. So if we get rid of this, different proteins can in fact act as markers. So when our body gets sick, when we contract a certain type of illness or disease, then there are certain cells that are more strongly affected, and there are certain cells that are responsible for fighting a given disease. And so when these cells fight the disease, they produce specific type of proteins that are designed to fight the given disease. And so basically, although we may not have any external symptoms for an illness, by measuring the levels of a certain protein in our body, we can identify whether or not we have contracted a certain disease. And so this is why, by measuring the amount of proteins, this can be a very important thing. Each disease may spark specific proteins to be produced within the human body. And if so, what we can do is we can test our bodies to see if there are a raised level of a specific protein. And if there is a raised level of or a raised amount of a specific protein, then we can see that we've contracted a certain disease. So not only can these protein markers therefore be used to identify illnesses, they can also be used to measure the effect of treatment. So if these protein levels are reduced, then it means the disease is kind of being fought off by our body. So we can measure the effect of treatment, and we can sort of just generally follow and keep track of the progress of a disease within the human body and how our body is fighting it off using measurements of these protein markers. Now, what we're going to look at now is an example. So we've drawn a small segment of a protein chain down here. So we'll say that this is a small segment rather than a full molecule in itself. And so we'll change this. So this molecule is extending off in each direction. So the first thing that we're going to do is we're going to write the abbreviated representation of the primary structure of this section of our protein. So the primary structure we know depends on the amino acids that form it. So first we want to identify the amino acids that have formed this section of our protein. So we've got two amide groups. We've got one here and one here. So we know that they are the joining parts, the parts that join different amino acids. So if we were to break this amide linkage here and this amide linkage here, then we could look at what lies between and figure out what amino acid we're dealing with. So if we look here, we can see that this is in fact the basis, or the leftover of the molecule glutamic acid. So if we were to get rid of this amide linkage and just have an amino group, and if we were to get rid of this amide linkage and just have a carboxyl group here, then we would in fact have a molecule of glutamic acid, and glutamic acid has the abbreviation GLU. Now if we look in the middle here, if this part of the amide linkage were in fact an amino group by itself, and this were in fact a carboxyl group, then this whole molecule here would be in fact a molecule of asparagine. So we represent asparagine using the abbreviation ASN. Now lastly, our last one is probably the most simple amino acid, is in fact glycine, or GLY. So now if we wanted to represent the primary structure of this section of our protein molecule, we simply write the three abbreviations, the three-letter strings that represent each amino acid in a row like this. So that is how we can represent the primary structure of this section of our protein. We've identified the three amino acids and we've written their abbreviations consecutively with hyphens between them. Now the next thing that we're going to do is we're going to look at the different bonds that can form to dictate the secondary and tertiary structure of this section of our protein. So we know that the secondary structure is caused by bonds, hydrogen bonds between adjacent peptide linkages. So what we've got here is we can see that we've got our NH, we've got a nitrogen bonded to a hydrogen here, and so that is so we've got a bit of a small positive charge towards this end due to the high electronegativity of nitrogen. Similarly, if we if we were to go here, we've got carbon double bonded to oxygen, and we're going to have a small negative charge at the oxygen due to its high electronegativity. And what that means is that this small positive charge and this small negative charge are going to attract each other. And so as this hydrogen and oxygen are pulled together, this is going to change the secondary structure of our protein molecule. Obviously, this can also happen in a lot of other places throughout the molecule. We've got another small negative charge there and another small positive charge here, and so on. And so we can get some more attraction going on across there in that way, and so on. And so that sort of bonding affects the secondary structure. Now, if we wanted to look at the tertiary structure, we can see that the carboxyl functional group of our first amino acid, of glutamic acid here, has in fact lost a hydrogen. So we've got a strong negative charge here. If we were to break these two amide linkages, we would have a glutamic acid ion due to the loss of this hydrogen atom. And so this is negatively charged here. Similarly, we've got our asparagine in the middle, and asparagine has acted as a base and accepted an extra proton, so it has a positive charge. So what happens now is that we have these two very strong, very strong electric poles now. So these are going to attract each other also. Now, because this is an attraction between the Z groups, this relates to the tertiary structure of our molecule. So that's just one type of bonding that can affect the tertiary structure. Now, if we wanted to look at other types of bonding that can in fact affect the tertiary structure, not only is it bonds between sort of ionic Z groups like that, we can also have the tertiary structure being affected by a few other types of attraction. So obviously, all these types of attraction refer to the Z groups of our proteins. So we've got again hydrogen bonds. In this case, we've got some ionic bonding. Although we're not actually dealing with ions, this is effectively ionic bonding, as our amino acids are effectively in ionic form as they form part of our protein chain here. We've also got a type of bonding called cross-links. So if we have some of our amino acids in fact contain Z groups with some sulfur atoms, and so what can happen is if we have a sulfur hydrogen bond bonded to the main part of our amino acid, then what can happen is if there is another sulfur hydrogen bond here, then these two sulfurs can in fact bond with one another directly by breaking off those two hydrogens, and we get what's called a disulfide link. And so because sulfur is present in some of the amino acids, disulfide links are in fact important parts of the tertiary structure. And lastly, of course, we can get dispersion forces between some of the very large Z groups present on our amino acids. So there are a few of the different types of forces that can affect the tertiary structure. So that is the structure of our proteins. They are very important to the human body. They basically form the majority of the matter and the structure of our bodies, and thus the study of these proteins and figuring out how they bond together is very important.